Okay, in this video I'm going to show you the process of transcription, and what you're seeing here is the nucleus, and inside the nucleus we have DNA, and it's just a certain portion that we call a gene, and we also have some free-floating nucleotides that are RNA nucleotides, and then we have RNA polymerase. So to start off transcription, RNA polymerase is going to go through the gene portion of DNA, and it's going to unwind the DNA. So it's going to look like that. What RNA polymerase also does as it goes through, and this happens pretty much at the same time, is it breaks the bonds between the nitrogen bases. So those hydrogen bonds get broken. Okay? And so it looks like this. And what we're going to do is use this top strand. That's our gene that we want to make into mRNA. So an RNA polymerase helps to do this as well. It brings in each nucleotide. So for right now, we're just going to bring them in. And if there's a T, it gives us an A. Then a U will come in next. Then a G. Whoops. Then a C. An A. A G. C, 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 and then A, U, and C. And so that makes our whole big long strand of mRNA. So they're going to bind together with hydrogen bonds, and just for the sake of time, okay, it's going to bond together, and it will look like this. One big solid strand. What that's going to do is then leave the nucleus. So let me just show you what this looks like. This comes out and it can fit through this hole in the nucleus. So it can leave. And that's called a nuclear pore. Then, with the help of the enzyme again, those bonds will reform, making a complete DNA strand again, and it'll retwist into its original form. Now, if this DNA tried to get out of that hole, you can see it's way too big. It doesn't fit. There's another one. It doesn't fit. So DNA can never leave. That's why you need this skinnier version, see, of the DNA. And the mRNA leaves. So it leaves the nucleus and it goes to the ribosome. Now this is the ribosome, and this is the start of translation. And these two processes occur not simultaneously, but one right after the other, because the mRNA that was made in transcription comes to the ribosome to do translation, which is to take the message that it brought from the gene in DNA and make it into a protein. So we're going to go ahead and see that this is the ribosome. The mRNA goes to the ribosome, and in the cytoplasm nearby, we have these tRNAs that are sort of free-floating. So our first codon, which is these three base pairs together, three bases, AUG. The tRNA that matches AUG would be UAC. That's going to come from the cytoplasm to the ribosome. It matches up, and it brings its amino acid. The next one is CAG, and CAG matches with GUC, and it comes and it brings its amino acid. What will happen is the amino acid from the first one gets dropped off, okay, and it attaches to the second one, and now this tRNA leaves to go pick up a new amino acid, and this moves down along with this on the ribosome, which leaves then space for the next one to come in, CCC, which is GGG. And then the amino acid chain again keeps building. This one leaves to go pick up another amino acid to be used again. This one slides down with the chain of amino acids still on it. And then we get to UAG, again, the amino acid chain shifts from the tRNA, sorry, I'm in trouble, to the next one. This one leaves. This one's here. A stop codon would be reached. This would be stop. And then that means this will get dropped off. This tRNA leaves. And these amino acid chains, this is the newly formed protein chain, will go off to another part of the cell, the ER, where it will be shipped then off to yet another part of the cell, okay, where it will be folded and repackaged into its correct form. This is the final protein. Okay, and that is the end of translation when we had that amino acid chain get shipped to the ER, which ships it to the Golgi, and that is our final folded form.